Assalamualaikum and greetings to Dr. Mahanem and Madam Nisa. So this is my week 10 progress report. So the first one is introduction. Ligno Biotech. Ligno Biotech Sendirian Berhad is a research-based biotechnology company that based in Balakong, Selangor. Their main focus is cultivation and commercialization of tiger milk mushroom Rhinoceros rhinoceros. However, there are other type of mushroom that are cultivated and commercialized in Ligno Biotech also. So the founder of Ligno Biotech is Dr. Tan Chon Seng and Ms. Ng. Next, company background. It was all started in 2002 where the International Convention on Biotechnology was take place. Back then, Dr. Tan and Ms. Ng was inspired by Dr. Tun Mahathir's speech. He gave speech on how he recovered from his chronic cough by taking tiger milk mushroom. So the first step, the duo do the identification of the mushroom that take a very long time, which is 6 months. Back then, there is no information on tiger milk mushroom scientific name. However, they're able to find them. So the duo went into the deep tropical forest and met with indigenous people to find tiger milk mushroom stock. So Dr. Tan and Ms. Ng took 8 years of trial and error and research. And finally, they managed to cultivate the tiger milk mushroom. Dr. and other expert scientists success in developing proprietary technology of Ligno in cultivating sustainable tiger milk mushroom. Besides then managed to cultivate tiger milk mushroom, Dr. Tan also discovered 2 species of Lignosus, which is Lignosus cameronensis and Lignosus tigris. And because of this, he was dubbed as the patriarch of tiger milk mushroom. After 8 years of dedication, finally, the moment of truth. In 2008, Ligno was founded with the support from Ministry of Agriculture, Malaysia. In 2009, Ministry of Agriculture also supported Ligno to set up plant for cultivation, promote and finally commercialize tiger milk mushroom. After success with tiger milk mushroom, Dr. Tan and his expert scientists also tried to cultivate Cordyceps sinensis, chaga, entrodia, and many more mushrooms that have health benefits. Later, in 2011, tiger milk mushroom listed in National Pharmaceutical Regulatory Agency (NPRA) as one of the active ingredients list. TMM product also has been registered with Drug Control Authority and Ministry of Health, Malaysia, and obtained registration code MAL in front of the code. And Ligno also has been certified as halal by JAKIM and organic by Scheme Organic Malaysia. Then, slowly, Ligno Biotech facilities has been certified to ISO 22000, HACCP and GMP, the standards for food manufacturing biotechnology company. And Ligno also awarded with BioNexus status. And today, the journey is still going on. Dr. Tan and his scientists are committed to Ligno's mission and vision. They treat this as a life patient to ensure humankind for better health quality. So here is Ligno's commitment. And now comes the most important part, my proposed project in Ligno Biotech. I wanted to do weekly monitoring of tiger milk mushroom, Cordyceps sinensis, Cordyceps militaris, and also entrodia. On my weekly monitoring, I will check on the growth and the conditions of these mushrooms. So when check the growth of the mushrooms, we can obtain many data. When checking the conditions of the mushrooms, I can check the contamination of any of these cultures. Regarding the checking the growth of mushrooms, different mushrooms have different growth rate. So during this monitoring period, I have to describe every week for each one of the culture. There are some mushrooms that are visible after 4 weeks. Some may take short period, and some may take a long period for the sclerotia to sprout or visible. From this, we could conduct correction and corrective action when there is no growth of mushroom within the time frame. Regarding the contamination check, contamination are caused by two types of microorganisms. First, fungus, and secondly, bacteria. Fungus indicated that the environment of the culture room is not optimal and promote fungal growth. Meanwhile, if the culture is contaminated by bacteria, that means the scientist responsible for pouring process is guilty. This may be caused by mishandling and wrong technique during pouring culture into the media. This is the display of 4 types of my proposed mushrooms. This is the objective of my proposed project. First, to describe the growth of the mushrooms. Earlier I said that there are many data that can obtain from describing the growth of the mushroom. Firstly, the data of the morphology of the mushroom from week to week. This can be used in the future reference. Second, the period it takes for the mushrooms to be visible on the culture also can benefit the marketing department. And lastly, how long does it take before it is ready to be harvested for the production scientists to do calculation before harvesting. The second objective is to check the condition of mushroom weekly until it is ready to be harvested. I wanted to inspect the culture for any contamination because contaminated culture need to be disposed following the SOP. The contamination are caused by fungus or bacteria. So here I show you the benefit that company can get from my project. The first is the information may become guidelines to Ligno. As for now, Ligno Biotech may already have a guide steps in the production area. Somehow, if I conduct the project, I may encounter a new problem that may require a new solution. So these new solutions can be integrated as a new guideline for the company. Second benefit is reference for other fellow production scientists. For example, after quality check test by QC and QA executive, results show a specific bacteria in the cultures. This can be overcome with implementation of correction and corrective action. As a result, the production scientists were reminded by QC and QA executive on the correct way and take considerations of all precautions. The third benefit, my information from the project can be useful for other departments such as marketing. Growth information from my project could be useful. For example, the 4 weeks that are required for the sclerotia to be visible. So, the marketing executive can wait for 4 weeks before the sclerotia can be visible and use the culture as display. My idea, I could integrate something from my project into the display of these species. I already thought about the idea of displaying each species according to their intervals of appearance changes. For example, I can prepare TMM for every 2 weeks to exhibit their appearance changes every 2 weeks until they are ready to be harvested. And lastly, the benefit from my project is for the new worker to refer when they are audited. This is the last part of my week 10 progress report. The brief description of activity, discussions, problem encounter, and troubleshooting. On the first week, Madam Nisa demoed us the use of potato dextrose agar, PDA. The PDA was used as media as it contained dextrose as carbohydrate source to stimulate growth of fungus cells. Potato in the PDA also is a base nutrient of most fungus, especially tiger mushroom. PDA with sterile tartaric acid 10% act as a selective inhibitor to allow fungus-only growth and inhibit bacteria. PDA also used as it is a suitable media for the fungi that grow to produce food. During subculture, take the outer mycelium cells only because it is younger and actively proliferate vegetative part, and it can last longer because it is young and new cells. Transferred cell media plate need to be wrapped with wrapper. This is to prevent dehydration. And one of the learned technique is when storing the culture in the incubator is inverted the top of the culture. This is to prevent condensation of liquid droplet on the plate lid and cause further contamination. This is my discussion of media preparation for mother culture. Different type of ingredient used is one of the result of Dr. Tan's extensive research, and this is Ligno's proprietary technology. Tapioca was added into the media for encourage sporulation of fungus. So when media prep, we are using the autoclave machine to sterilize the media. So the bottle cap of Duran shot bottle is loosened prior to autoclave to prevent microbes enter by pressure differences. Bottle cap is also loosened to prevent the explosion because of the pressure differences and allow adequate steam penetration when autoclave. Autoclave tip also used to mark the bottle that has been autoclaved. When we have done autoclave, we take the media and use the technique of leaning the bottle 45 degrees and shake to allow the cloudy concentration part to dissolve. And this is also to ensure thorough media without any cloudy part. My second week, I have done subculturing of mycelium cell TF6, red yeast Malaysia, jelly white, and koji green. And this is my discussions of the subculturing. When subculturing in the laminar flow class 2 biohazard safety cabinet, the positioning is very important. The clean culture needs to be arranged with the most clean one that put far inside the laminar flow. This is to prevent any spores in the air near us to fall into the new culture. Secondly, practice pushing culture plate with finger without holding the plate as a whole in our hand. This is to reduce possibility of contamination also. In my third week, I also learned how to prepare media for chaga and cordyceps sinensis. Media preparation started with arranging PP box and fill it with rice as media. Then add RO water and MP11 powder, the Ligno secret powder, before mix gently. Finally, autoclave the media at 121 Celsius for 30 minutes. For media preparation, when filling RO water in the PP box with rice media, water is made sure to go through the PP box wall. This is to ensure no MP11 powder stick at the wall of the PP box. Autoclave. This is my brief description for week 4 activities and discussion. From week 5 until week 8, I was heavily involved in the production department. The last part is problem encounter during the 8 weeks of training and troubleshoot. First, the sieving machine. The problem is that sometimes the rubber band that are used to seal the sieving net are wear off. So to overcome it is I have to add thin rubber band to hold the main rubber band to its place. Next, mixing machine. I was unable to handle the mixing machine yet. Most of the time Daniel would handle it and he would supervise me when I handled the machine. The mixing got a tiny straight line aperture at the cover of the machine. So, to prevent the aperture from letting the raw materials to fall off, I need to close the hole with masking tape. And this is the picture of the failing machine. This is the most problem-prone machine that I have encountered during my 8 weeks of. So that is all for my week 10 progress report. I wanted to thank to Dr. Mahanem, Prof. Sheila, and all lecturers in biosciences and biotechnology. Thanks for the chance for me in the Ligno, and I promise to do my best for UKM and for my future. Thank you.